Despite the incredible technological advances in the various imaging modalities and treatment options available to all cancer patients, cancer is still a significant public health problem and the second leading cause of death. Even though we're in the midst of a COVID-19 crisis, by the end of 2020, cancer would have taken about 10 million lives with 15 million new cases in accordance with the World Cancer Reports. Some forms of cancer or brain tumors, such as glioblastoma multiforme or simply GBM, really has a very poor prognosis because it is very aggressive, highly radioresistant, with a low sensitization profile. My research in the physics department at Creighton University intends to use semiconductor nanoparticles or quantum dots to fight these highly resistant forms of cancer. Now, research has demonstrated emphatically that the presence of high electron density, high atomic number nanoparticles within a tumor volume greatly increases the production of secondary electrons, which in turn greatly enhances the production of highly reactive chemical species such as ROS or reactive oxygen species, which in turn hinders DNA replication and thereby stopping cell or tumor growth. In our previous published work in peer-reviewed journals, we had demonstrated that the presence of ROS in solution decreases the fluorescent intensity of quantum dots. Now, using a novel assay as well as a specially made device that I built in the lab, we were able to radiosensitize highly resistant glioblastoma cells as indicated by the fluorescent intensity curve. As you can clearly see, the red curve is the fluorescent intensity of glioblastoma cells irradiated in the presence of quantum dots. You can see that the drop in the fluorescent intensity is an indication of an enhanced production of highly reactive oxygen species or ROS within the given volume. An increase in ROS production hinders mitotic cell division, which stops cell growth. This has great clinical implications. We can inject a given tumor volume with biocompatible pegylated quantum dots, then irradiate this tumor volume. The presence of quantum dot increases the local dose, and there, because the effect is short range, we will spare the surrounding healthy tissues. This will mean that the tumor complication probability will increase and the normal tissue complication probability will greatly decrease, and thereby increasing the therapeutic ratio with a very small dose. Thank you so much for listening to my talk.